All right. I think it's recording. So, yeah, thanks for agreeing to talk. Take it away, Shesha.
Thank you so much. Hello, everyone. I want to start off by thanking the organizers for giving me this wonderful opportunity and for organizing this wonderful seminar. My name is Shesha, and I'm a PhD student in the group of Professor Massimiliano Esposito at the University of Luxembourg. And today I'm going to present work I did with Francesco Avanzini and Massimiliano, and you can find the reference here. And so I want to talk to you about asymptotic growth in chemical reaction networks. Now, obviously, the first question is, what do I mean by growth? And by growth, I just mean the accumulation of an observable in the system. And in the physicochemical context, this is very, very common. For instance, two processes that immediately come to mind are polymerization, where if you start with a bunch of monomers, you can end up with very, very large polymers. And the second example that comes from biology is like the cell cycle of unicellular organisms, where you have an initial cell that grows and divides. If you, for instance, if you take the E. coli cell cycle and you measure the total amount of proteins, and you can actually see that there are regular patterns, for instance, in the cell cycle, that the number of proteins increases exponentially in the time between divisions. So with this notion of growth, it's also true that a lot of people have looked at growth before and in the context of chemical reaction networks. And so I want to discuss a couple of models that people have used. Again, this is not exhaustive. I've just made a selection. But it sort of illustrates the point that I want to make. And more specifically here, I'm going to think about concentration growth. So we have a chemical reaction network, and we are thinking of an ideal dilute solution. And we are thinking in the deterministic regime. So there are no fluctuations, and the concentration is the dynamical variable. And people have wanted to understand how the concentration scales with time. One example of where people have looked at this is non-enzymatic template replication. That's a very fancy name for saying that you have a template molecule, which is written as C here, and you have precursors A and B. And basically, there is an autocatalytic, it's autocatalytic after the three steps. Basically, you have an autocatalytic reaction in which the template replicates itself. So you see the entire reaction network here. You have C binding to A and B, it dimerizing, and then breaking into two molecules of C. And for reasons which I won't go into here, it's interesting to ask, how does the concentration of C scale in time? And you can see that, I mean, this is not very hard. You can do an analysis with the ODEs, and you can see that it actually scales parabolically in time under a few assumptions. A second example, and this goes back to the physical example I gave you in the first slide. This is called the Hinshelwood cycle. And Hinshelwood wrote it down to model cell replication. So you have X1, X2, and X3. Forgive me for the super bad drawing, but like this is the sort of way in which I sort of show this. And each of these species, X1, X2, X3, you can think of them as some coarse-grained set of molecular species inside a cell. So X1 could be like the total amount of protein, for instance. And what you say is that, so you have this mutually catalytic cycle. So X1 helps produce X2, X2 helps produce X3, and X3 helps produce X1. And people have used this to study, for instance, not just the growth rate, but if you looked at the stochastic regime in this, although I'm not personally interested in my work, but if you looked at the stochastic regime in this example, you could, for instance, look at things like growth fluctuations using these models. So my interest in growth comes from a slightly different perspective, and that is from the perspective of thermodynamics. In my group, we are very interested in non-equilibrium thermodynamics. And the basic idea is that, of course, we all know the theory of equilibrium thermodynamics. And it's a very powerful theory. It's something that we all learn in our undergraduate studies. And the point is that it allows us to evaluate notions of efficiency and energy costs, but in transitions between equilibrium states. So this is a very, very restrictive assumption. And of course, going beyond this assumption of equilibrium is quite hard. But over the last 30, 40 years, there has been a lot of progress in stochastic thermodynamics, which is at what is called the mesoscopic level, where fluctuations play a great role. And there has been a lot of progress. I will not refer to this, but you can look up the literature if you're interested. And in stochastic thermodynamics, there are frameworks to go beyond just equilibrium. And you can start thinking about dissipation, and you can start thinking about the free energy cost necessary to sustain certain complex behavior. And in my group, a lot of what we focus on is going back from this stochastic regime to the deterministic regime and thinking about non-equilibrium thermodynamics in this macroscopic regime. So, for instance, an example of this could be something like chemical oscillation, like the Belousov-Zhabotinsky oscillator, or pattern formation, which are manifestly non-equilibrium processes. You cannot do this at equilibrium. And the question would be, how much free energy do I need to sustain a pattern? Or how much am I dissipating in this oscillation? And we have a framework, which I will present shortly, to allow us to sort of understand this free energy cost. And the idea I want to ask is, can I use this to write down, like, to do the same analysis for growth that I was talking about in the last slide? But here there is a problem. And the problem is that the framework that I will show you later needs every reaction to be elementary and reversible. And all the models that I showed you, they involved irreversible and coarse-grained reactions. Of course, I'm not saying that that is wrong. I'm just saying in that modeling framework, of course, it's quite apt and useful to use coarse-grained and irreversible reactions. But from a thermodynamic perspective, that makes it very, very hard to accurately estimate the energy costs. And so this actually leads you to a first question, right? When is growth possible in a chemical reaction network with all reversible reactions? And here, I want to make the point that transiently, it is always possible to grow. So I'll come back to this at the end. But of course, if you take a chemical reaction network, which is autocatalytic, and you start with a small amount of the autocatalyst, of course, you will have an initial exponential increase in the concentrations of the autocatalysts. This is definitely true. But today, I want to focus on something slightly different. And this comes from the work of Francisco, my colleague, who is also a co-author on the paper. And Francisco was studying this metabolic network, and he was studying what we call an open chemical reaction. So you have a chemical reaction network here, which I will describe now. And basically, it's a very simple network. You have like a fuel species F, a waste species W, a substrate S, and a product P. And you have this E, EF, EW, E star. They're just enzymatic species, basically. So E combines with F to form EF, which can exist either as EF or EW. And EW can give you E by releasing W. Similarly, EF can take up S to give the complex E star, and E star can go back to EW by releasing the product P. And the reaction network that Francisco was studying had F, S, W, and P being exchanged with reservoirs. Now, I will in more detail describe how this exchange happens later. But for the moment, it's enough to just say that when you open this network, he saw two regimes of behavior. In the first, you reach fixed points, and and we call these fixed points as non-equilibrium steady states. And in the second regime, what he saw was this kind of a behavior, which I plot on the right-hand side, where this is concentration in some rescaled units versus time in some rescaled units. And you see that there is a continuous accumulation of F, and here I think you have F and S, whereas W and P are sort of held constant. And this we call asymptotic growth. And now, of course, the restrictions that I told you, the problems I told you before don't apply, because every reaction here is reversible. Although I've drawn it like this, every reaction here is reversible, every reaction is elementary, and we know how to deal with exchanges. So in open chemical reaction networks, what we have now is this idea that you can enter a phase where you have this continuous accumulation. And the question that we wanted to answer was, how does this happen? What is the dynamics of this growing state? And I motivated the full thing by saying thermodynamics. So is there a cost for growth? How much does it cost you to grow? So in the next 10 minutes or so, I want to talk about the basic formalism necessary. And in the last 10 minutes, I present my results. This slide is about chemical kinetics, and I think this audience is very, very familiar with it. But just to set the notation and the basic stage, I will use the Michaelis-Menten as a sort of example throughout my slides. And just to set this thing again, you have two reactions. You have an enzyme that reacts with the substrate to form the complex ES, which then breaks into the enzyme and releases the product P. Typically, I'm thinking about S and P being exchanged with reservoirs. And the way we write this reaction mathematically is using a vector of species, E, ES, SP, and then you write down these vectors nu plus rho, nu minus rho. And every reaction you can write down as a product of nu plus rho dot alpha, giving nu minus rho dot alpha. So what the news measure is the number of each species participating in a reaction. The difference between the nu minus rho and nu plus rho gives a column of what is called the stoichiometric matrix. So, and the stoichiometric matrix tells you in each reaction how much of each species you're either consuming or creating. Keep in mind that every reaction is reversible. So even if I choose a forward direction arbitrarily, inverting it doesn't change any of the results. So in the first reaction, you're consuming E and S to create ES, basically. Coming to the kinetics, we are of course interested in how the concentrations evolve. We collect the concentrations into this vector Z, and we split the vector Z into two subvectors, X and Y. So the X species, they change only due to the concentrations change only due to chemical reactions, and this you can see here. The species E and ES, they change only due to reactions. Whereas the Y species, which are S and P, they change due to reactions and due to exchanges. So if you write down how the reactions evolve, you get this set of coupled ODEs. And Sx and Sy are just submatrices of the stoichiometric matrix. The current itself, we model using mass action. So every reaction has a forward flux and a backward flux, and both fluxes follow the law of mass action. So the forward flux of the first reaction would be proportional to the product of the concentration of E and S. So far, I told you how the chemistry happened. Now I have to specify the exchanges. So we studied three mechanisms of exchange. The first one we called concentration control, and this is the most typical thing that you would see. And the idea is that you fix the concentration of the Y species. So this either happens because you assume that there is sufficient Y species that no reaction happens. Basically, that any, I'm sorry, not that no reaction happens, but that any sort of change in the Y species due to the reactions is negligible. Or you assume that there is a reservoir and and this reaction is very, very quick. So effectively, you can treat the concentration of the Y species as constant. So in this example, the concentration of the S species is constant and the concentration of the P species is constant. And of course, here I refer you to this reference if you want a physical setup which sort of achieves this. But there are other setups wherein you don't need to fix the concentrations. And in fact, you can think about fluxes. And one such setup is what is called the tank reactor, where you have influxes of material and outflux of everything inside the reactor. We sort of abstracted this and said, okay, we can have influxes at a constant rate, which is exactly as what happens here. But the outflux rate, we sort of took two different choices. If the outflux rate also we model as constant, we achieve what is called flux control, where you have an influx and an outflux. So in this example, you have a constant influx and a constant outflux. And the other example is where you have a constant influx, but the outflux is proportional to the concentration of the Y species. So in this example, you model the outflux as a matrix B acting on the current concentration. And in this example, for instance, the P species is being pulled out at a rate proportional to how much P is inside. Okay, so this is about the exchanges. And now I want to talk about the thermodynamics. For lack of time, I will not go into details. I will just remind you that we are thinking of ideal dilute solutions. And in this case, what one can do is you can formulate quite rigorously a notion of thermodynamics by defining chemical potentials and free energies. I'm not showing you their expressions because I don't think it does anything important. But what we really need is just this. So G here is the Gibbs free energy, and it's the thermodynamic potential. And you can write it as mu. Mu is the chemical potential of species alpha times the concentration. The second term here is what is called the chemical work rate, and it is measuring the rate at which free energy is coming in due to the exchange with the reservoirs. And the third term here is what we call the entropy production rate. It measures the total dissipation in the system and the environment. And the entropy production rate is always positive, and you can see the formula written down here. So this also illustrates why we need elementary reversible reactions. You see that the entropy production rate involves both the forward and the reverse fluxes for each reaction. So neglecting even one of these would give you a nonsensical result, basically. How do you think of the second law? Well, for the moment, you can just think of it as a sort of balance equation. WC is the rate at which free energy comes in from the surrounding. What a part of that free energy either gets stored into the Gibbs free energy or it gets dissipated. And if you want to know more, I refer you to the literature here. And just for an example, in the Michaelis-Menten here, the rate at which the free energy comes in would be mu SIS, which was because of this, and mu PIP. Second thermodynamic notion that I need is the notion of an equilibrium and a non-equilibrium steady state. So if we have a closed chemical reaction network wherein there is no exchanges, and you start with whatever initial concentrations, you will always evolve towards a state called the equilibrium. In this state, there is no current in any reaction. So it's not just that this current and this current are equal and they cancel each other. It is that the current in each reaction is zero and the dissipation is zero. I sort of plot that here when you have energy versus time, and here you just have time. And you see that both the entropy production and the Gibbs free energy go to zero. On the other hand, if you open a chemical reaction network, generically you can reach a fixed point, which is called a non-equilibrium steady state. And this is an example that I show here. In this case, the concentrations reach a fixed point, so they don't change in time, but there will be a chemical current. And the way you see it is like this. For instance, in this example, you can think of some amount of S coming in from the reservoir, reacting with the enzyme to form the complex ES, and then splitting back to give E and P, and the P that was produced goes out. So you still have a net flux going from S to P. And because this reservoir is at some chemical potential mu S, and this is at some chemical potential mu P, what you're doing is you're creating a current between two reservoirs at different chemical potentials. This has a dissipative cost, and that is precisely what your entropy production is telling you. And in fact, again, you can, I plot it again, and you see that this time the blue dotted line, which is the entropy production, goes to a non-zero constant. Okay, so now that I've set the complete stage, I want to go back to the example that I told you with Francesco, and here I want to specify that he was studying mixed control. So he was saying that the fluxes F and S are being injected in, while W and P are being extracted with a rate proportional to how much W and how much P is inside the system. And here he saw two regimes. One, depending on the initial condition, you can either reach a non-equilibrium steady state, or you reach the state where things asymptotically accumulate, and what we call growth. So before I go into my discussion of my results, I sort of want to discuss this point of why we sort of say that. So when we define growth formally, we say that if the system keeps doing this, eventually the concentrations reach infinity. And this infinity thing, I understand, is a bit strange, but it's just saying that this is the asymptotic time scale. I'm saying that this is not the transient, it's the asymptotic. Of course, there are other physical mechanisms that we are not considering that would kick in after different time scales, and we are not modeling. So infinity should not really be seen as infinity, but it should be seen as saying that this is the asymptotic result. Just as a refresher, because I will keep referring to these terms, concentration control means you fix concentrations. Flux control means the influx and outflux rates are constant. Mixed control means the influx rate is constant. The outflux is proportional to how much is inside. Okay, with this, I go to my results. We systematically analyzed different classes of chemical reaction networks. And there was one class which we could treat analytically, and those we call dynamically linear. And as an example, it's unimolecular and pseudo-unimolecular chemical reaction networks. And in dynamically nonlinear networks, which are multimolecular networks, we sort of studied different examples. For dynamically linear networks, you can prove that there is no growth in mixed or concentration control. And you can show that growth happens in flux control. So for dynamically nonlinear networks, we sort of proved by examples saying that growth is possible here and here. That is, we found examples where growth happens here and growth happens here. And if you recall already, Francisco had shown this, so this is not that surprising. So what we did was unveil the mechanism by which this happens. And in the interest of time, because I have only 10 minutes left, what I want to do instead is to sort of talk about an example from here and an example here, just to illustrate the mechanism and the thermodynamic implications of growth. For the full details, again, I would refer you to read the paper. So the first example is a unimolecular network under flux. And unimolecular, I've been using the term. It just means that it's a network of unimolecular reactions. So every reaction has this form, x1 to x2, x2 to x3, and so on. You can draw the reaction network as a simple graph. And in this case, as an example, I'm showing you one in which I'm pushing in x4 and I'm pulling out x3 at constant rates. So in this setting, it's not, if you think about it, it's not really surprising that there is growth, right? Because you can say that, okay, I'm pushing things in at a constant rate, I'm pushing things out at a constant rate. If I push things in faster than I pull things out, of course, I'm going to accumulate mass. So that is true. But what is interesting is the thermodynamics here. And just to show you, for a closed unimolecular network, the rate equation takes this form, where now it's a matrix equation. You can solve it exactly, and you can show that Z reaches an equilibrium state. This equilibrium state is a right null eigenvector of W. But in flux control, the equation gets modified as W plus a constant vector. And now you can show that there, in fact, what happens at every time T is that Z is some Z equilibrium of T up to a constant. What do I mean by this? So at every time, you can sort of decouple this into two processes. A process by which I'm putting in and taking out mass through the reservoirs, and the chemistry, which is interconverting whatever mass is coming in. So if I stop the influxes at any time, the influx and the outflux, if I stop it at any time, the system will relax to an equilibrium with the amount of mass that is already in the system. So this is why you have a Z equilibrium of T, because what you're doing by pushing things in faster than you pull them out is that you're putting more and more mass in the system. And this is sort of pushing the equilibrium state to larger and larger concentrations. But what the chemistry is doing is that it is sort of, by reactions, it is sort of reorganizing the mass into different species such that you're always close to this equilibrium state. So notice that the equilibrium state is scaling proportional to time, but you're at most a constant up. This means at very, very large times, the state of the system is indistinguishable from the equilibrium state. And as an example, here I show you a plot of the concentration versus time. And you see that in this case, the equilibrium is when x1 is equal to x2 is equal to x3 is equal to x4. And you see that it's actually, that thing is actually happening, that they are equal to each other and they're moving away. So what is happening is that the influxes push in a bit of x3 and pull out a bit of x4. But the chemistry reorganizes the net mass that you've gained into x1 and x2 so that the ratios are effectively hovering around 1. And this, and so you're growing, but in a way that you're always close to an equilibrium. So we call this equilibrium growth. And as a thermodynamic consequence, you can actually analytically show that the entropy production in this case goes on decreasing and actually goes at 1 over time, which means at large times your system is not dissipating. Any free energy you're putting in as work is getting converted into the Gibbs free energy of the system. So it's like very, very efficient growth. But a very non-trivial growth mechanism can be seen in the second example. And here I will again use the Michaelis-Menten. And this is like, this shows the same phenomenology that Francisco was showing, where and here what I'm doing is I'm pushing in S and I'm pulling out P. And in this case, depending upon the rate at which you push in S and the initial condition of the system, you can either have this growing phase again, where here you see S is growing, or you can sort of, or you can reach a non-equilibrium steady state. And how do we analytically show this? Well, here we do a thing where we say that the reactions inside are very, very quick. And so we can assume that the species E and ES are always at a steady state. And then we can coarse grain out to write an effective evolution for SN. And in that effective evolution, you then ask when can it grow with a linear rate, for example. We chose the linear rate just from being inspired by the simulations. And if you do that, you see that the expression that you get for the rate is that the velocity at which the S species grow is related to the rate at which it comes in minus an initial condition dependent thing. So this thing I'm not introduced, but it's an initial condition dependence. And the physical mechanism for this is very interesting. It's basically what is happening is that because the amount of enzyme in the system is constant, the rate at which S gets converted to P has a bottleneck. So there is a maximum rate at which S can get converted to P. Now, if this were not there, you can easily imagine a scenario where S comes in, is converted to P, and P goes out. And so you always maintain a steady state. But if, because the amount of enzyme is fixed, there is a maximum rate at which S can get converted to P. And now if you pump in S at a faster rate than this, then you will have growth. And this is the dynamical underpinning of growth in these mixed control. And this is why you're getting this initial condition dependence. Depending upon the amount of enzyme, you can either have growth or a non-equilibrium steady state. And here I sort of show it. Here I sort of show you this initial condition dependence in plots. The quantity I'm plotting on the y-axis is like the rate of change of the amount of S in the system versus time. And you see that when the initial condition is not favorable, you always reach this quantity always goes to zero. Eventually, it reaches a steady state. Whereas here, in this example, you see that this quantity is positive and you have it reaches a non-zero rate of growing. So dt, so the time derivative of the amount of S is positive, which means S is increasing continuously in time. Finally, to wrap it up, I have to also tell you about the thermodynamics. And of course, the thermodynamics here is out of equilibrium. And you can compute everything exactly in this example, and you can show that you are actually growing far from equilibrium, where you're paying, again, you're paying huge costs, basically. So again, the thing is that to sustain this growing state, you have to pay a huge amount of chemical work, and a lot of that is being dissipated. So in a sense, this is very inefficient. So I want to quickly summarize in the last few minutes. I showed you guys something about asymptotic growth, which is a regime that we showed can happen in open chemical reaction networks when there are influxes of species. And hopefully I convinced you that this is a non-trivial phenomenology, and that the structure of the chemical reaction network determines whether you have an equilibrium or a non-equilibrium. Of course, the thing that I was saying about the structure, you can make more precise. Like I'm sure all of you are familiar with conservation laws, and using conservation laws, you can make this very, very precise. But in the interest of time, I just choose to give you the most basic message. To wrap it up, I should also mention perspectives. At the start, I mentioned transient growth, and here, of course, we come to the work of Alex Blockowitz and David Lacoste and Philip Engay, who sort of classified autocatalytic reaction networks into these motifs, and then the follow-up by Philip Engay and Jeremy Unterberger, who showed that autocatalytic networks with small enough concentrations can have an exponential rate of increase. And so these are examples of what I would call as transient growth, and understanding them is also very, very important. And the second example, which I want to highlight, is growth with volume. What I showed you was concentration growth, but in many examples, you might want to think of a reaction network embedded in a volume, and the volume is growing along with the number. And this, for example, again, I think we had a talk about this already this year by Kamimura, I think by Atsushi Kamimura. So yeah, with this, I want to stop here. And thank you so much. Once again, thank you to Alex and Praful for giving me this wonderful opportunity.